Picture this. It's February 1953, and two young scientists, James Watson and Francis Crick, are sitting in the Eagle, a cozy pub in Cambridge, England. The air smells of ale and wood smoke. Francis Crick, a talkative 35-year-old with a booming laugh, leans forward and declares to anyone who'll listen, "We found the secret of life." His partner, Jim Watson, a lanky 24-year-old American with long hair and a mischievous grin, feels a twinge of embarrassment at the boast. But deep down, he knows Crick might just be right. They've just built a model, a twisting, elegant double helix that explains how life's blueprint, DNA, works. But how did they get here? Let's rewind. Our story begins decades earlier, with scientists slowly piecing together clues about DNA. Back in 1869, a Swiss doctor named Johann Miescher scooped some gooey stuff from white blood cells and called it nuclein. He had no idea he'd stumbled on DNA, the molecule that carries our genetic code. Fast forward to the 1940s, and an Austrian biochemist named Erwin Chargaff makes a curious discovery. In DNA, the amount of adenine always matches thymine, and guanine matches cytosine. It's like finding that every recipe in a cookbook uses equal parts of certain ingredients. Weird, right? But nobody knows what it means yet. Meanwhile, across the Atlantic, the legendary chemist Linus Pauling is cracking molecular puzzles like nobody's business. He's already figured out the spiral structure of proteins, and he's got his sights set on DNA. Pauling's a rock star in chemistry, and everyone expects him to solve this one too. But DNA is tricky. It's a long molecule, like a twisted ladder, and nobody can figure out how it's built or how it carries life's instructions. Enter Rosalind Franklin, a brilliant scientist working at King's College London. She's a master of X-ray crystallography, a technique that uses X-rays to capture shadowy images of molecules. In 1952, Franklin and her student Raymond Gosling take a photograph that will go down in history, Photo 51. It's a blurry, cross-shaped image that screams helix to anyone who knows how to read it. Franklin's meticulous, but she's cautious. She's not ready to shout helix just yet, because another form of DNA she's studying looks less twisty. Plus, she's stuck in a tense lab with Maurice Wilkins, who thinks she's his assistant when she's very much her own boss. Their frosty relationship, marked by misunderstandings and clashing egos, slows progress at King's, creating an opening for outsiders like Watson and Crick to swoop in and synthesize the data. In 1951, Jim Watson, a young American fresh out of grad school, attends a conference in Naples. He's there to learn about viruses, but his life changes when he sees one of Maurice Wilkins' early X-ray images of DNA. It's not as clear as Photo 51, but it's enough to haunt him. That picture, Watson later writes in his memoir, The Double Helix, I could not forget. He ditches viruses and heads to Cambridge's Cavendish Laboratory, where he meets Francis Crick, a physicist turned biologist who loves to talk, argue, and dream big. The two hit it off, united by a burning question: what is DNA's structure? Despite their enthusiasm, they face skepticism from established scientists. Take Erwin Chargaff's visit to Cambridge in the summer of 1952. Over drinks at Peterhouse, Chargaff listens to Watson and Crick's ambitious plans, then mocks Watson's long hair and American accent. He grills Crick on base chemistry, and when Crick admits he can't recall details and says he'd look them up, Chargaff dismisses them as ignorant pretenders. Watson later recalls how Chargaff's contemptuous smile still stung him weeks later in Paris. It was a humbling reminder of their gaps in knowledge. Now, here's where it gets juicy. Watson and Crick aren't exactly chemistry experts. In fact, Watson's knowledge of organic chemistry is so shaky that he picks up a textbook from a street vendor, thinking it'll help him crack the code. With this bargain bin book, they build a model of DNA, a triple helix with the bases sticking out like awkward decorations. They're so excited, they invite Franklin and Wilkins to see it. Big mistake. Franklin takes one look and points out a glaring flaw. The chemistry's all wrong, and the water content doesn't add up. Turns out, that street textbook was outdated, and Watson's model is a disaster. Their boss, Sir Lawrence Bragg, is so embarrassed he bans them from working on DNA. Ouch. But this flub shows you don't need to be a genius from day one to make history. Watson didn't know much chemistry, but he learned by doing, by messing up, listening to critiques, and trying again. That's science in action. As he writes in the Double Helix, after Franklin dismantled their model, he realized we had to know more chemistry to get it right. The rivalry intensifies with Linus Pauling, who sketches his own triple helix model in late 1952, inspired by electron microscope images but lacking top-notch X-ray data. There's a myth that Pauling could have beaten everyone if not for his passport troubles, denied in early 1952 due to his left-wing activism against nuclear weapons. After protests, he gets a limited passport and visits England, but he sticks to protein lectures and skips King's College, missing Photo 51 entirely. So, the delay didn't really cost him the discovery, it was his focus elsewhere. By early 1953, Pauling publishes his model with phosphates awkwardly crammed in the center. Watson and Crick spot the flaw right away. Those negatively charged groups would repel each other like same-pole magnets. It's a dud, but they know Pauling will fix it soon, lighting a fire under them. Bragg lifts the ban to avoid another loss to the American giant, and the Cambridge duo dives back in. Help comes from unexpected corners. Their office mate, Jerry Donahue, a chemist from Caltech, corrects a key mistake. Textbooks showed the DNA bases in the wrong form, enol instead of the more stable keto, which messed up hydrogen bonding. This tweak is crucial for the base pairs to fit. Another colleague, mathematician John Griffith, chats with Crick about quantum mechanics and complementary surfaces for gene replication, suggesting adenine thymine and guanine cytosine might pair up, reinforcing Chargaff's data and planting seeds for the breakthrough. Then, a series of lucky breaks and sneaky moves changes everything. In January 1953, Watson visits Franklin at King's, pretending to warn her about Pauling's paper. The meeting turns explosive. Watson pushes for a helical model. Franklin snaps back angrily, and she chases him out of her lab. But during the chaos, Wilkins shows Watson Photo 51. Watson's jaw drops. The cross pattern screams double helix. He later admits in his memoir that seeing it without Franklin's consent was pivotal. Adding to the ethical gray area, Max Perutz, Crick's collaborator, shares a King's report with Franklin's measurements. Unmarked as confidential, but she's unaware. Historians debate if it was a breach, but it gives Watson and Crick the numbers they need. Back in Cambridge, Watson's playing with cardboard cutouts of DNA's bases, adenine, thymine, guanine, cytosine, like a kid with puzzle pieces. One February morning in 1953, he's shuffling them around and bam, he notices something incredible. Adenine fits perfectly with thymine, and guanine pairs with cytosine like molecular puzzle pieces. The shapes match, and the pairs explain Chargaff's equal ratios. Crick jumps in, figuring the strands run in opposite directions, twisting with 10 steps per turn. They build a metal model, and it's beautiful. Two sugar phosphate backbones spiraling around each other, with base pairs as rungs. Crick's wife, Odile, an artist, even draws the iconic double helix diagram for their paper. They invite Franklin, Wilkins, and Bragg to see it. This time, no one laughs. Franklin nods. It fits her data. At the Eagle pub that afternoon, Crick's secret of life boast echoes through the room, and Watson feels the weight of what they've done, though he later admits the phrase might have been embellished for dramatic effect. In April 1953, Nature publishes 3 papers: Watson and Crick's model, Franklin and Gosling's Photo 51 data, and Wilkins' supporting evidence. Watson and Crick's paper ends with a humble line. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Understatement of the century. Their model explains how DNA replicates, passing life's instructions from one generation to the next. It's the dawn of modern molecular biology, but the story doesn't end without some drama. In 1962, Watson, Crick, and Wilkins win the Nobel Prize, but Franklin, who died of ovarian cancer in 1958, gets no mention. She's ineligible because the Nobel isn't awarded posthumously. Her draft notes show she was close to the Helix herself, and today she's hailed as an equal contributor. Watson's memoir, The Double Helix from 1968, spills the beans with gossip and humor, but draws fire for portraying Franklin as mean and priggish and admitting to using her data without consent. Critics say it oversimplifies, but Bragg praises its raw honesty, letting readers feel the researcher's struggles. And let's not forget Linus Pauling's class act. When he inspects the model in Cambridge, he graciously says, "You had the answer," despite his disappointment. At a Solvay conference, he publicly admits his model was wrong and lauds theirs, later winning the Nobel Peace Prize for his anti-nuclear activism. His sportsmanship shines amid the cutthroat race. So what's the takeaway? The discovery of the DNA double helix wasn't just about X-rays and chemistry. It was about people. People who made mistakes, like Watson's dodgy textbook model. People who clashed, like Franklin and Wilkins in their tense lab dynamic. People who took risks, like Watson and Crick sneaking a peek at Photo 51. People who faced setbacks, like Pauling's passport drama that almost, but didn't, derail him. And people who learned on the fly, like Watson with his street-bought chemistry book. It's a reminder that science isn't just lab coats and equations. It's curiosity, rivalry, ethical dilemmas, and a bit of luck, all twisted together like a double helix. If you love this story, please like and subscribe, share it with a friend, and let us know what other discoveries you want to hear about. Until next time, keep asking questions, and maybe check the copyright date on your textbooks.